you want to get your phone out to look at the live chat? It started.
Okay. Zishan, where's that one with the? That's the one, yeah.
To hide it, yeah.
Yeah, found it. Okay, are we live?
Yes, we are live. We are live.
Oh, amazing, amazing.
Welcome,
Let me just quickly give us a minute, guys. Okay. Just this is the first one out of the how many we got? 12 live streams.
No, not 12. I think 10 in total.
Is it? Oh, 10. My bad. Yeah, 10 live streams. Okay. So, oh, bro, we hit 80k subscribers on YouTube. Come on, Zishan. Whoa, smashed the 80k, guys. This year I want to hit that 100k subscriber so we can get that little. What would you get when you get 100k subscribers, Zishan?
Plaque, YouTube plaque.
You get a plaque. Okay, guys. Wow, already 2k viewers. Come on, guys, you're smashing it. You guys are smashing it. I'm gonna sniff this to. Oh, if you know about this, if you know, you know. In fact, let me give it another one. Quick, another sniff. Guys, comment if you know what this is. This is a magical thing that will clear your brain and make you focus 10 times more. I wish I had 100 of them. I'll give you all one, but unfortunately, I've only got one. So I can only use it myself, but it's okay. Comment if you know what this is and from what country you get this from. I can't even read the language there. Okay, so guys, welcome to the first of many live streams, but in particular, welcome to the GCSE Biology Paper 1 live stream by Examqa. Now look guys, what we're gonna do is, we're going to make sure that you smash that paper tomorrow. Me, Zishan, and all these moderators sitting in the back who don't want to be on camera because they're very embarrassed. We're going to make sure that your exam paper goes well tomorrow, okay? Now, give us 1 minute. We're just quickly sorting out something, okay? But in the meantime, if you have friends, yeah, I know some of you have friends, go tell your friends to join this, okay? And don't be a gatekeeper, because remember, guys, gatekeepers, they don't make it anywhere in life. If you want to be a gatekeeper, you're going to lose out. So go tell your friends, say, look, the live stream started. We're about to go and smash biology. Now, by the way, there are 3 types of people in this live stream right now. 1 second.
Working for you, yeah?
Just my things. Okay, we'll start in.
10k likes will start.
We'll start when we hit 10k likes.
Yeah,
That's what Zishan said.
Let me check double check what the likes are right now.
Yeah, Zishan, where's my thing?
Okay.
Dropbox.
So we're on 100 likes. Okay, that's not enough. We need. Okay, let's see. Abdurrahim, how many likes are we on?
Likes? 184.
Okay, 1k likes and we're gonna start.
All right, we're gonna start in 1k when we hit 1k likes. Come on, guys, get ready. Get ready. We got 3k viewers. Come on, everyone.
We need 1k likes and we'll start. Just for the algorithm.
To smash that algorithm, we're gonna need to. Yo, Zishan, quick thing here.
It's not working.
I'm gonna have to.
Abdurrahim, you don't need to plug too much just yet, yeah? I'll tell you when to plug proper.
Where's my thing? So guys, look, we're going to be covering combined science and triple science, yeah? But the way we're going to be covering it is, we're going to do all of combined science first. So if you're a triple science student, you should still be in this because the topics we're going to go over is still going to be coming up in your exam, okay? And then we'll have a small Q&A and then we'll start the the triple specific topics, okay? Now, it will be recorded, okay? So the live stream is being recorded. So that means you can view it later as well. But I would say still try to be here now because at least this way you can ask me questions if you got any questions, okay? All right, we're going to start in 5 minutes. Get ready, everyone. We're just going to start in 5 minutes.
Are we at 1k likes yet?
I believe we are very close to starting. Shout out. Okay, guys. Let's have a shout out from which part of the planet are we watching the live stream? Because I know for a fact there's one kid in Thailand who goes to Shrewsbury Academy next to the river that's watching this. I know that for a fact. Which part of the planet are we at right now, guys? Southampton, Dagestan. Bro's from Dagestan. Okay, fair enough. Where else have we got? UK, Tunisia, Libya. Okay, Zishan.
Yep.
Tell me, if they haven't, what's it called? If they haven't revised biology, what's gonna happen today?
Okay, if they haven't revised, okay, look guys, if you have not revised biology, we need to
make that a bit
big one for
bigger.
this, yeah.
Yeah.
Okay, guys, this is serious now. We need a big one for this. Okay, if you have not revised biology for the 5 years you've been in secondary school, year 7 to year 11, 4 years, it does not even matter, because tonight you're going to witness a masterclass, and even if you're on a grade, sub 5, sub 0, you're going to become a grade 7, 8, or 9. And if your foundation, you're going to become a grade 5.
Amazing.
This is guaranteed. This is tried and tested. Every year we've been doing
Zishan,
this.
should they take notes or just listen?
Okay. Brief notes, not detailed. Brief notes, not detailed. So it's mainly for understanding. If you're writing notes, you're missing what Mohammed's writing,
Yeah.
you're doing the wrong thing.
Okay.
Okay, because it's going to be intense.
Brief.
Very intense.
Keep it brief but light. Yeah.
Yeah.
All right. Fine. Can we start?
Give me 1 second.
Oh, can you fix this?
Oh, yeah, yes.
All right. Okay, guys, get ready. We're gonna start in 30 seconds. Get your notes ready. Go tell your friends who haven't joined. They're gonna miss out. Every moment of this is gold. Okay, so here we go. All right, we're just waiting for the green light from the moderators at the back. Guys, stop wrestling. Why are you wrestling already? This is not gonna be. All right, here we go, guys. So, countdown. Starting in. Shout out if you go to Willesden Green. Shout out if you go to Leytonstone. All right, guys, get ready. One more thing. Let me just tell you one more thing, okay? This is personal advice from me to you, because I've done exams as well, okay? Make sure that tomorrow, before your exam, you don't speak to anyone. Just go straight to the exam, do your exam, and then when you finish your exam, don't speak to anyone and go home straight away, okay? And I'll tell you why later. But that's one request from me. And we've officially hit 4K viewers. All right, Zishan, we're going to start then, yeah?
So, can you start? Let's see.
Yeah? All right, here we go. So, okay, everyone, here we go. So we're going to start now. Let's do it. First topic that we're going to go over is microscopes. So, first of all, in terms of microscopes, and guys, I am going to be going a little bit quickly, okay? So I want you to pay attention to the speed that we're going through. So we're going to start with just a quick structure of the light microscope. So we've got the eyepiece lens, okay? And the eyepiece lens by itself actually has ×10 magnification, yeah? Then we've got the objective lenses. These are the little ones over here, okay? And they can have various magnifications. So ×10, ×100, ×1000, yeah? Then, the reason this is called a light microscope is because light passes through the light source, through the specimen, and then into your eye, okay? So that's the reason it's called a light microscope. Now, a couple of more things. You got the slide, yeah? That's where we put the specimen on, and you've got the stage where it all gets clipped onto. Now, the most important part of the microscope are the two knobs. So you've got the coarse knob, or sometimes referred to as the rough knob, and the job of that knob is to move the stage up and down closer to you, okay? That's the job of the coarse knob. The other knob is called the fine knob, okay? And the job of that one is to make the image clearer. So make sure you don't confuse those two. All right, so for example, if you've got a blurry image, yeah, you move the fine knob to make the image clearer, okay? So don't confuse those two knobs between them, okay? Now, moving on quickly. One sec. So this part is very important. With microscopes, you'll definitely get a question where you're going to have to either calculate magnification, image size, or actual size, okay? So let me quickly show you how to do that. So for example here, we can see that we have a specimen, okay, which has produced an image in this microscope. And the formula that you guys need to know is magnification equals image size divided by actual size, Okay, that's what the formula that you need to know. That's some noise coming from there. So here's the question here. For example, if it says that this cell in the image has an actual diameter of 20 micrometers, calculate the magnification. So here's how you do a question like that. Number one, you're going to obviously take your formula, magnification equals image over actual, okay? Now, some of you might not know what's the image and what's the actual. So let me tell you the clue. If you have to use your ruler to measure it, that is called your image, okay? The actual will be given to you in the question, or sometimes it's basically the smaller one. So for example here, to calculate my magnification, they've given me the actual size already in the question, okay? To get the image size, I'm going to have to get my ruler and physically put it on your exam paper and measure the length of that cell, okay? So for example, 5 centimeters. So I know my image is 5 centimeters and my actual is going to be 20 micrometers, which is given to you in the question. Now, this is the part where everyone makes a mistake. Before we continue, we have to make sure that both of the units are the same. So right now, one of them is in centimeters and the other one is in micrometers. You have to make them the same. It doesn't matter which one you make them to, but they have to be the same, okay? So here's a quick table for you to compare so you can see how you move. So for example, let's convert centimeters into micrometers. So we're going to start from here, centimeter, and we're going to go all the way to micrometer. So that means we're going to times it by 10, and then we're going to times it by 1,000. So we're going to times it by 10,000, okay? So keep this table saved, so that way you can always refer back to it later. So look, so we go from 5, we times it by 10,000, that gives us 50,000 micrometers. Okay, so remember, the image size is the one that you use your ruler to calculate. The actual size is usually given to you in the question, and then you just do magnification equals image over actual. Okay, everyone? Now, sometimes you might get a question that looks like this. Can you see over here, in the question, they didn't give us the actual size, but instead, they give us a scale bar, okay? So, to calculate magnification for a question that has a scale bar, all you have to do is just calculate the length of the scale bar. So let me show you what I mean by that. So again, magnification equals image over actual, yeah? But this time, instead of using the ruler to measure the length of the cell, we're going to measure the length of the scale bar using our ruler. Okay, so let's say I use my ruler, I measured the length of the scale bar, and it said 3 centimeters. That's my image, right? Because I use the ruler. So I know the image is 3 centimeters, and I know that the actual is 15 micrometers. Yeah. So you put them into the fraction, and then remember you have to make sure that both of the units are the same. So we're going to take our centimeters and we're going to convert it into micrometers. Remember, like before, times it by 10 and then times it by 1,000. So 3 times by 10,000 gives you 30,000. There you go, okay? So that's how you do it. So in summary, if you get a scale bar, forget about the picture, go straight to the scale bar, measure the length of the scale bar, and that will tell you the image size of the magnification. Okay, guys? Good. That's the first topic. Oh, actually, hold on. A little bit more left, okay? So very quickly, now there are 2 types of microscopes that we have to know in GCSE. You have the light microscope and the electron microscope. So a very quick comparison about the pros and cons of these 2. Okay, so here we go. First one, let's start with the light microscope. And let's talk about some advantages. So number 1, it provides a color image. Okay. Number 2, you can view living specimens. So if you put the specimen into the microscope, it won't have to die. Okay. Number 3, it does not require lengthy preparation. In other words, you can just say it's very simple to use. And number 4, it's much more cheaper. So these are the 4 advantages of a light microscope. Now, disadvantages are, it has a lower resolution compared to the electron microscope. It has a lower magnification, and you can't see small organelles. So for example, if they ask you, Josephine wanted to see the mitochondria, can she use a light microscope? No. To see the mitochondria, chloroplast, or ribosomes, you have to use a electron microscope. Okay, guys? Good stuff. All right, so that's the first topic out of the way. Let's go into a very, very important practical, which is the osmosis practical. Here we go. All right, next topic. So osmosis. Now, what I'm going to do, guys, this is quite a long practical. So what I want you to do is just listen, and at the end of it, I'm going to give you a summary that you can just copy. Okay? So here we go. So first of all, osmosis, we all know by now, is the movement of water across a semi-permeable membrane. Make sure you mention the word semi-permeable. If you just say movement of water, you don't get the mark. Okay? Now remember, if you look at this picture over here, we can see that one side has more water. Okay? So that side we call the dilute side, or you can say that it's the side that has a higher water concentration compared to the side on the right. Okay, and we know that water goes from an area where it's dilute to an area where it's concentrated. Now, a very important thing about osmosis is that it is passive, which means it does not require energy, just like diffusion. The only thing that requires energy in biology is active transport. Okay, cool. So anyway, we're going to do a practical to find the water potential, or sometimes they could say the sucrose concentration of a vegetable. So in this case, we have a potato, but it could be a carrot, it could be any other type of vegetable. Okay, so like I said before, listen to the practical, and I'll give you the summary at the end of it that you can just copy and paste into your exam. Here we go. So step one is we're going to grab the vegetable, again our potato, and we're going to cut out some cylinders. Okay, after that, after we cut out the cylinders, we're going to use a mass balance to measure the mass of all those potato cylinders. Now, when you measure the masses of the potato cylinders, it's okay if they don't all have the same starting mass. That's completely fine, because we're going to be using something called percentage change. Yeah? So if they ask you, should we worry about having the same mass of potatoes? No, it's fine. Don't worry. We're going to calculate percentage change later. Okay? So here we go. Here's our potato cylinders. Now, once we've done that, we're going to prepare sucrose solutions. So here we have beakers which have water and sucrose. And notice that they have different amounts of sucrose. Okay? So the one on the left has the most amount of water and the least amount of sucrose. And the one on the right has the most amount of sucrose and the least amount of water. Okay, so we're going to put our potato cylinders into those beakers. Once we do that, we leave them in there for the same amount of time. So you let the potato cylinders inside the beakers for the same amount of time. Over time, what will happen is, look at this little picture. Okay, ready? Watch. You see that? The potato cylinders get bigger and smaller. Can someone tell me in the chat, why are the potato cylinders changing size? What's going on? How come some of them are getting bigger and some of them are getting smaller? It's because obviously of osmosis, okay? But anyway, moving on with our practical. We take out the potato cylinders, and then this part is very, very important, okay? Before you reweigh them, you have to pat them dry. Give them a pat on the back. But don't rub it too hard, okay? Because if you pat it too hard, you're going to remove all of that water. So just a very, very, very light tap, like the one that you give to your friend after school, yeah? All right, so once you give it a little pat, you remove the water, okay? Then you're going to reweigh those potato cylinders. All right, now some of them are going to become a bit more chunkier and some of them are going to become thinner. But don't worry, the next step is to calculate percentage change in mass. Now, if you don't know how to do that, don't worry, I'm going to show you right now. All right, here we go. So to calculate percentage change in mass, all you have to do is do the final minus the original divided by the original. That's it. Okay, so for example, if my potato went from 5 grams to 8 grams, all I have to do is do 8 take away 5 divided by 5. Yeah, and then that gives you, and don't forget obviously to times it by 100 because it's a percentage change, and that will give you your percentage change, so plus 60. Okay, so remember, in percentage change, if it's a plus, it means that it has gained mass, and if it's a minus, it means it has lost mass. Okay, cool. Once you've done that, then we're going to plot a graph. Okay, so we're going to plot a graph of sucrose solution of those different beakers that we had versus percentage change in mass for each potato in that beaker. Okay, when you plot your graph, you're going to notice something. You're going to notice that it looks like this. Okay, so just look at my points on my graph. Yeah, some of them have gained mass and some of them have lost mass, which is completely fine. But what you're going to do next is you're going to join all of those lines together with a line of best fit. Okay, now remember, what was the original question in the practical? It was calculate the sucrose concentration of my potato, right? So to do that, here's what you do. Very easy. You go to your graph, you go to the part where there is no change in mass, okay? So what number is that? What number does it have no change in mass? looks like it's 0.6. That is the concentration of your potato. Okay, why is that the concentration of potato? Because at that concentration, the potato didn't get bigger or smaller. It means it was the same concentration as that sucrose. Okay, now remember, this was a very, very long practical. Here's what you need to know for your exam. So just copy this out. Look, 8 points. Screenshot this, save this, or later on, just come back to this part and just memorize it. So if that practical was long for you, don't worry, just memorize this, put it into your exam, and you're good, okay? And obviously, if they use a different vegetable instead of potato, if they use like aubergine, yeah, then change it to aubergine, all right? Cool. Now, one more thing. How do we make sure that our practical is, how do we make sure that it's a fair test? Here are 4 ways to make sure that it's a fair test. Number 1, when you cut out this potato cylinders, use the same potato, okay? So get all the cylinders from the same potato. Number 2, make sure that all of the potato cylinders are left in the solution for the same amount of time. Number 3, make sure that the temperature of the solutions are the same. And number 4, make sure that the volume of the solutions are the same. Okay, so very easy 3 marks or 4 marks if they ask you, how do you make sure that your potato cylinder, how do you make sure that your practical is a fair test? All right, and finally, 2 more things, okay, very, very important. In your practical, make sure that when you put the potato cylinders into the water, it's fully submerged. And again, don't forget to pat them dry at the end. All right, good stuff, guys. That is the osmosis practical out of the way. Let's move on to the next topic. Let's keep it going, everyone. Okay, so next topic coming up. 1 sec. Here we go. Is there a football match today? Oh, someone's talking about Liverpool on the side. Ooh, okay. Do we have any Arsenal fans in here? Oh, fair enough. Oh, okay. So all the Gunners won, yeah? All right, let's do it. So now we're going to go over stem cells. A very quick and very short topic, but just a good refresher for you guys. All right, here we go. So stem cells are unspecialized cells or undifferentiated cells, okay? So what that means is they don't have a specific job. However, they can turn or they can differentiate into any specific type of cell. That's the cool thing about them. Okay, now where do we get stem cells in animals? So in animals, they're found in 2 places. Number 1 in the embryo, okay, and number 2 in the bone marrow, so inside the bones. But the thing about them is that the embryonic stem cells are much more versatile than the ones in the bone marrow, okay, and that's because they can turn into any type of cell. Do you guys understand that? Okay. Cool. Whereas the stem cells in the bone marrow can only turn into blood cells, like red blood cells and white blood cells. Okay, so make sure you don't confuse that part. All right, now, because embryonic stem cells can turn into any type of cell, it makes them very useful in medicine. Why? Let's look at a couple of reasons why. Number 1, they can be used, okay, so write down for me 3 reasons why embryonic stem cells are useful in medicines. Okay, here we go. The first one, they can be used to replace faulty cells. Yeah, very straightforward. If an animal has faulty cells, they can be replaced by a healthy stem cell. Number 2, they can be used to treat diseases like diabetes by making insulin-producing cells, okay? That's that one. And number 3, so for example, if a person is paralyzed because their nerve cells aren't transmitting information properly, we can give them new nerve cells and treat paralysis. So that makes them very, very special. Okay, everyone? And number 4, stem cells are not rejected by the host. So for example, if you get organ transplant from another person, that person's antigens will trigger your immune system. But if you get stem cells transplanted into you, your host cells are not going to attack them. Okay? Good stuff. Now, this word over here, therapeutic cloning, just refers to treating someone's diseases using stem cells. Okay? So that's what that means. Now, what are some of the disadvantages of stem cells? Here are a couple. Number 1, obviously we know that stem cells have ethical issues associated with them, okay? So if you're using stem cells, that means you're also risking destroying potential life. Number 2, they can be contaminated by viruses, so even though you're using them to treat someone, that stem cell could be infected. Okay, now here are 2 arguments that you need to know for and against the use of stem cells. Let's start with for the use of stem cells. So scientists that use stem cells, they say that, oh, number one, oh, this stem cell was obtained from an unwanted embryo. It was going to die anyway. We just used it for scientific research. And number two, we're treating serious diseases with stem cells, so the benefit outweighs the cost. Okay, that's what they say. And the people that are against it, this is their argument. Number one, that you're killing potential life, and number two, that you could use alternative treatments instead of going straight to stem cells. You could use other treatments. So make sure you remember both sides of the argument. Okay, finally, plants also have stem cells that are found in the shoots and in the roots, okay? And those areas are called meristems. And one special thing about them is that they are found throughout the entire life of the plant, okay? From birth to death, they're always there, okay? And what's one good use of stem cells in a plant? We can use them to clone identical copies of that same plant to prevent extinction. All right, nice. Stem cells out of the way. Well done, guys. You guys are doing very well. Keep locked in. We're going to go straight to the next topic. Here we go. Lishan, how's everyone doing?
Yeah, it's good. You know,
What's the general consensus? Anyone?
They're very tame this year. They're not really making oil jokes.
Oh, fair enough. Very locked in, yeah?
Yeah, very locked in.
Lovely stuff. Okay, guys, so here we go. We're going to quickly do an easy topic, but just as a reminder, let's quickly do some food tests. Here we go. So number one, if you have a sample and you suspect that it might have protein. Now, protein is obviously, we know, fish, chicken, egg, that's protein. But another type of protein that they could test you with are antibodies and enzymes. Those are also proteins as well, okay? So if you add the burette reagent to proteins and protein is present, then it will go purple, okay? So remember, burette is blue, so B for burette, B for blue, and then P for protein, P for purple. So protein goes purple. But if there's no protein, it just remains blue. Okay, everyone? So burette for protein, if there's protein, it goes purple. If not, it remains blue. Yeah? Next one, iodine. Iodine, okay, is used to test for starch, which is found in potato, rice, etc. Now, iodine itself is orangey brown, but if you mix it with starch, it goes bluey black, yeah? But if there's no starch, then the iodine remains orange brown. Easy one. Okay, next one. Lipid. Now, there are two for lipid that you need to know. One is Sudan 3, okay? So when you add Sudan 3 to lipid, you form two layers, and the top layer goes red if there's lipid. So lipid is fats, yeah? But if there's no lipid, the whole thing goes red. Yeah, so Sudan 3, lipid, two layers, top layer goes red. The other way to test for lipids is using ethanol. So what you do is you add ethanol to the sample, then you shake the sample, and then you add water. Okay, and then if there was lipid, it would go cloudy. That's called the emulsion test. Okay, so that's the other one for lipid. And finally, for sugars like glucose, we use Benedict's. Now with Benedict's, again, B for Benedict's, B for blue, it stays blue. But if there is sugar, then you have traffic light. So remember, with a traffic light, what you have? You have green for safe, less sugar, orange for a bit of sugar, and then red if there's loads of sugar. Okay, and if there's no sugar, it stays blue. There you go. So that's the food test. I know that's a very easy one, but a lot of you might have forgotten the color. So here you go. That was a good reminder for all of you. Let's move on to the next topic. Everything okay, Dish?
Yeah, there's no big break.
All right, cool. We're going to do two more topics and then we'll have a short little break. Okay, everyone? Here we go. Let's go to the circulatory system. Who knows, while this loads up, who can tell me the name of the blood vessel that takes blood out of the heart towards the lungs? Who can tell me the name of that one? In the chat, go. If you know, let's see if you guys know it. What is it called? Ooh. I just saw 10 people say the wrong answer. In fact, they're still saying the wrong answer. Guys, what are you doing? Okay, fine. Okay, so someone just said veins. I gave up. Oh, here we go. Let's do it. The circulatory system. Okay. So first of all, remember that our heart is a double circuit. Okay. So what that means is it pumps blood to our lungs. Blood that's deoxygenated, which means it doesn't have oxygen. Okay, it pumps it to the lungs, where the blood becomes oxygenated, so it picks up oxygen. It comes back to the heart. That's one circuit. Okay, that's one circuit. Then it pumps blood at the same time to the rest of your body, Okay, oxygenated blood to the rest of your body, where the oxygen gets used in respiration and then deoxygenated blood returns back to your heart. Okay, so you have two circuits happening at the same time. That's why our circulation system is called a double circuit. Okay, but if we zoom in deeper, let's have a look at the actual structure of the heart. So first of all, the heart, obviously, in your exam, they usually flip the picture of the heart. So you have the right side on the left of your paper, and then you have the left side on the right of your paper. But once I saw in an exam that they flipped it again. So if you're not sure, just look at this part right here, okay? The part that's thicker, the part that has a thicker wall, that's always the left side of the heart. Just remember that, okay? Cool. So here we go. Now, we have vessels that bring blood into the heart, okay? Now, whether it's green, whether it's blue or red, whether it's oxygenated or deoxygenated, doesn't matter. If it brings blood into the heart, it's called a vein, okay? So the vena cava is bringing blood from the body, and the pulmonary vein is bringing blood in from the lungs, okay? That then moves into these little chambers over here, which are called the atria, yeah? So you have two atria. Then, from the atria to the ventricles, we have tiny little valves, little flaps that are there to prevent backflow, okay? Those flaps are called atrioventricular valves. They're the ones that prevent backflow from the atria to the ventricles, okay? Next, we have the bigger chambers, the ventricles, okay? And then finally, so blood goes in like that, it goes back up. So in, out. Now, look, we have a set of other valves over here. These are called the semilunar valves, okay? So we have the atrioventricular valves, which are between the atria and the ventricles, and then we have the semilunar valves, which are between the ventricles and the arteries, okay? So these two arteries, this one is called the aorta, which takes blood to the body, okay? And what kind of blood is it? It's oxygenated blood to the body. And this one, which was my original question, it's the pulmonary artery. That's the vessel that takes blood out of the heart towards the lungs. So if you guys got that right in the chat, well done. You're not cooked for tomorrow. And if you got it wrong, now you know the answer, so you're not cooked as well. Let's go, guys. Come on. All right, next part. We know that all our muscles are controlled by our brain. Yeah? For example, if I want to move my biceps, if I want to move my body, I think about that and it happens by itself, okay? But can you stop your heart from beating? Can you think and stop your heart from beating? Come on. It doesn't work, right? Why? Because your heart doesn't listen to your brain. It has its own little brain called the pacemaker. And that pacemaker, another name for it, by the way, is SAN, which is your sinoatrial node. The pacemaker is found in your right atrium, okay? And its job is to control the heartbeat, okay? Now, some people, their pacemaker gets damaged. So what that means is their heart starts to beat irregularly. What do we do with that person? We give them a artificial pacemaker. So it's like a little machine that gets surgically implanted into their heart, and that helps to control the beating of the heart. Yeah? Now, with this person, you have to make sure that they completely avoid big magnets, yeah? Such as the MRI magnet. Why? Because if they go into an MRI with an artificial pacemaker, it will just rip it out of their heart. Completely rip it out of their heart because of the metals in there. Yeah? So that's a very important precaution of the artificial pacemaker. All right. The next part I'm going to go through very, very rapidly, okay? So listen to this, guys. We're going to quickly compare the three types of blood vessels, artery, vein, and capillary, and their layers. And you just need to know what each layer does, okay? Here we go. So first of all, the hole in the middle of the blood vessel is called the lumen, okay? That's the name of the hole, the lumen. The layer that covers the lumen, or the first layer, is called the endothelium, and it's always one cell thick, okay? Now, the next layer, okay, is smooth muscle. Why do we even have muscle in our blood vessel? What's the point of that? Does anyone know why we have muscles? Two reasons. Number one, it's to stop the blood vessels from exploding because of all that high pressure. But the second reason is because those muscles help your body to control which part needs more blood during different times. I'll give an example. Yeah, you know when you have a big fat meal, you get a food baby, right? What does that mean? It means that your body has moved all of the blood towards your digestive system. Okay? Or for example, when you go to the gym and you get a good pump in your biceps, why does your bicep suddenly swell up? It's because those muscles in your blood vessels have suddenly moved all the blood towards your biceps, okay? So the muscles are there for, one, to prevent explosion of your blood vessels, and number two, to redirect blood flow, yeah? Okay, and then finally you have the last layer, which is collagen, and again that's there as the outer layer to help to strengthen your blood vessel, okay? Now, let's quickly compare arteries and veins, because although they have the same 4 layers, the differences are 3. Number 1, veins have, okay, so arteries have a higher pressure compared to veins. Why? Because the blood pumps directly into an artery. So naturally, arteries always have a higher blood pressure, okay? That's one difference. The second difference is that arteries have thicker muscle compared to veins. Again, that's there to prevent, to allow the artery to withstand that high pressure. Moving on. The next part is, because of all that thick muscle, the arteries have a smaller lumen, okay? Much more smaller compared to the vein. And the last one is that the vein has a valve to prevent backflow, whereas the arteries does not have a valve, okay? So let me summarize it one more time. High pressure in the artery, thicker muscle in the artery, smaller lumen in the artery, and then vein has valve, artery does not have a valve. There you go. Blood vessels completed. Let's go, guys. Next topic. Okay, Zishan, we'll do one more topic and then we'll have a 2-minute break, yeah? Okay, good stuff, good stuff, good stuff. Let's go, everyone. Come on. Guys, if you're liking the live stream so far, give us a thumbs up. Okay, we'll move on to the next topic.
Someone's asking, is it Examqa or UCR?
Listen, guys, once I finish the important topics, I'm going to open the chat to you guys. Whatever topics you want me to teach you, I'll teach you as well, okay? But for now, just listen to the ones that we need to, the more important ones that we need to go through. Okay, here we go, here we go. One sec. All right, here we go. Ready? Next practical, measuring photosynthesis. Okay, now look, we all know what photosynthesis is, yeah? We know that it's when carbon dioxide and water makes glucose and oxygen. We all know that, yeah? But how do we measure how quickly photosynthesis is happening? So to do that, we're going to do this practical. Again, there should be a summary at the end of it, okay? So I'll let you look at the summary. But for now, listen to this, yeah? So here we have a special aquatic plant. It's a plant that lives in water. It's called a pond weed, okay? So we're going to allow this pond weed to do photosynthesis. In other words, it's going to produce what gas again? What gas does it produce in photosynthesis? Oxygen, okay? And we know that the more photosynthesis it does, the more oxygen is produced, and this bubble over here that we have in the capillary tube is going to be pushed further along, okay? So all we're going to do is we're just going to measure the distance that the bubble travels. If the bubble travels further, it means the pondweed has done more photosynthesis. Okay, that's literally it. So step 1, we're going to place a pondweed in water and connect it to a capillary tube and a gas syringe over here. All right. Next, step 2 is we're then going to place a white light source near the pondweed at a specific distance, okay? Because what we're going to do is we're going to investigate the effect of light intensity on the rate of photosynthesis. So you place the white light near the pondweed, okay? Then what we're going to do is we're going to measure how much that bubble has traveled over a given amount of time. So let's say we give it 10 minutes and we measure how maybe the bubble has traveled 3 centimeters over 10 minutes, okay? And then we know that the distance moved by the bubble is proportional to the rate of photosynthesis, okay? So those are the 4 points that you want to mention in your answer. Now, when you're doing this experiment, you have to make sure that outside factors do not mess up this experiment, okay? So here's what you control. Here are some control variables that you have to keep the same when you do this experiment. The first one is you have to make sure that the temperature of the experiment is the same. So the pondweed test tube has to be placed in a water bath and give it the same temperature each time, okay? Number 2, you want to make sure that the amount of carbon dioxide that's inside this test tube, right? Because there's carbon dioxide in here that the plant uses, right? But that carbon dioxide comes from a special liquid called sodium hydrogen carbonate. sodium hydrogen carbonate. So you want to make sure that the amount of sodium hydrogen carbonate inside that test tube is the same each time, so the plant gets the same amount of carbon dioxide. All right, obviously we know the third one is the time left for photosynthesis should also be the same in each experiment. And finally, this part's very important. We have to make sure that we use an LED light source, not a filament lamp. Why? Because LED light does not produce heat. It only gives out light, okay? Good stuff. Now, the next part is the inverse square law. It's a quick little calculation that I will teach you, and it will help you to calculate by what factor the light intensity has changed, okay? So look, let me show you. So light intensity is proportional to 1 over d squared, okay? And I'll show you how to calculate d. Let me give you an example. So to calculate d, all you're going to do is you're going to do the final distance divided by the initial distance. That will give you d, okay? And then all you got to do is 1 over d squared. Let me show you with the practical. Here we go. So imagine it said that the pondweed was 20 centimeters away from the light initially, okay? So that was our initial distance, 20 centimeters. Then the light moved closer. So now the light is 5 centimeters away, okay? So it went from 20 to 5, yeah? So remember, how do you calculate D? You're going to do final over initial, yeah? So what was our final? It was 5. What was the initial? 20. So that's going to give us our D. What is that going to be? Let's calculate it very quickly. 0.25, okay? So that's how you calculate D. And then all we have to do is we're going to do 1 over D squared, okay? So 1 over 0.25 squared. Put that into your calci. Take your calci tomorrow and put that into your calci. And you're going to get 16. What does that mean? It means that when we move the pondweed from 20 towards 5 centimeters, okay, the light intensity increased 16 times. Does that make sense? So that's basically how you do it. Let's do one more just to make sure you guys lock in with this one, okay? Here we go, one more time. So this time, the pondweed was 32 centimeters away, okay? And then it moved forward to now being 4 centimeters away. So it's come very, very close. So we know that the light intensity is going to be really high. But by what factor has it changed? Let's do it. So remember, what was final? 4. What was my initial? 32. Okay, so 4 over 32 is equal to 0.16, I believe. Let's see. Oh, my bad, 0.125, yeah? So we did 4, our final, divided by our initial, and that gives us 0.125. That's the D, okay? And then all you have to do is 1 over D squared. 1 over this number squared, and that gives us 64. So when we move the light from 32 to 4 centimeters close, being close, the light intensity increased by a factor of 64 times. That's it. All right, guys. Good. So that practical is done. Let's move on to the next practical. Let's do this, guys. All right. What? We hit 80K subscribers, yeah? Ooh. What if we hit the plaque this year? That would be man. What should you? We should have a challenge. If you hit the plaque, we'll do something crazy. All right, guys. You ready? Respiration and exercise. Very quick one, okay? Not too long on this topic. Just a very quick reminder. So first of all, we know that energy comes from glucose, right? When it's burnt inside the mitochondria. Or when it's not burnt, I should say, when it's used to provide energy. So we know that already. Mitochondria uses glucose and it releases energy in the form of ATP, okay? Now, once energy is produced, what does our body do with that energy? What does it even do with the energy? Here are 3 things that your body does with the energy produced from respiration. Number 1, it uses the energy to join small molecules like amino acids into larger molecules like proteins, okay? That's a very important one. So that helps you to grow. Number 2, it allows your muscles to contract, okay? So without energy, your muscles will not be able to contract, so you can't move your biceps and triceps. And number 3, the most important one, in warm-blooded animals, it helps to maintain their body temperature. So if you don't respire, your body temperature will fall. Okay, so that's very important. Now, we know that there are 2 types of respiration. Okay, what are they again? Aerobic and anaerobic. Let's quickly summarize both of them. Here we go. So aerobic means respiration with oxygen, and anaerobic means respiration without oxygen. Okay, so let's start now. Aerobic respiration is the same, doesn't matter what you are, whether you're an animal, whether you're a plant, fungi, it's the same. Okay, but anaerobic depends on if you're an animal, Okay, or plants and yeast. They have different reactions for anaerobic respiration. All right, so here we go. Very quickly. So aerobic, what was it again? It was glucose. And guys, remember the formula for glucose. C6H12O6. Remember that one, yeah? Glucose and oxygen becomes carbon dioxide and water, yeah? And remember, what's the most important part about this? Energy is being released. Okay, so anaerobic in animals, this part, so aerobic is the same for all living creatures, yeah? But now anaerobic in animals, it's a little bit different. Okay, here we go. So glucose, but because it's anaerobic, no oxygen. So what happens to the glucose? What does it turn into? If it's inside an animal, it turns into big L, lactic acid. Okay, of course you get a bit of energy, but not as much as aerobic respiration. Now, what does lactic acid do? It gives you muscle fatigue, pain, and cramps. Okay, it's toxic. But now, in plants, anaerobic respiration, the glucose doesn't turn into lactic acid. Okay, in plants and yeast, glucose turns into ethanol, which is an alcohol, and carbon dioxide. Okay, so it turns into something completely different. Now we have to know what these are used for. Okay, so look, ethanol can be used to make like chemicals like beer, antiseptics, medicines, etc. Okay, whereas carbon dioxide can be used to make bread rise. Okay, so if you ever seen flat bread, okay, and then you have big loafy bread, the reason it's loafy and big is because there's carbon dioxide releasing into the bread, making it rise. Okay, so this reaction has another name, by the way. It begins with F. It's called fermentation. Okay, so that's fermentation. All right, good stuff. Now, two very important things that happen to your body during exercise. One that happens to your heart, and the other one that happens to your lungs. Okay, so your heart rate increases and your breathing rate becomes deeper and faster. Why does this happen? 2 marks. The first one is because the reason your heart rate increases is because you want to get as much oxygen to your cells and you want to remove as much carbon dioxide from your cells. Okay? So during exercise, your heart rate and breathing rate increases to get as much oxygen from the air into your lungs, and then from your lungs to your cells. And do the opposite with carbon dioxide. Okay? All right. Final part. When we do anaerobic respiration, okay, that means respiring, burning glucose without oxygen, our body goes into something called an oxygen debt. So what that means is there's a buildup of lactic acid because we used glucose without oxygen. So how do we pay back the oxygen debt? How do we pay it back? By, when you finish exercising, your heart rate and your breathing rate continue to stay high for a while, okay? And that's how you pay back, that's how you drive in oxygen to your cells, okay? And that removes all of the lactic acid. Now, lactic acid, 2 things happen to lactic acid. There's 2 ways that lactic acid gets removed. One of them, we said that it reacts with oxygen, okay, and it turns into carbon dioxide and water. That's one of the ways lactic acid gets removed. The second way is that it goes to the liver and gets turned back into glucose, okay? So most of you probably knew the first one, but the second one, that one, make sure you know that as well. Those are the 2 ways that lactic acid gets removed from your body. Okay, everyone? Good stuff. All right, so we're going to have a little break for 2 minutes, and then after that, we'll come back and finish off the rest of the topics. Okay, here we go.
Okay, boys and girls, very, very well done. That is very impressive, the concentration you guys are having. As you can tell, this is a very intense live stream, right? And it's designed to be like that. I mean, if you're revising the day before, you know, you can't really expect to get a light live stream, right? We're going to be covering so much stuff. So that's why we mentioned maybe it's best not to have like detailed notes. Take very brief notes because your mind is going to be doing most of the working today. It's mainly for understanding. If you can understand these concepts, then tomorrow you're going to be likely to put it into action. But if you're making detailed notes and so on and so forth, it's the day before the exam. Let's be real. Like, you're not going to be able to digest everything. So the main things you understand, that's what you want to focus on, okay? Okay, my moderator, if you, my main moderator, if you can put the main messages in the chat. Guys, we've got something very special for you guys, okay? Firstly, I want to say this live stream is the first of many live streams, okay? So you need to subscribe so you can keep up to date with all our live streams. For example, on Thursday you have Maths Paper 1. So Wednesday night we're also doing a Maths Paper 1 live stream, okay? So subscribe so you can keep up to date with all the time changes and so on and so forth. We're doing this for all exams. So if you're finding this useful, you know, tune into our next live streams. I'm going to tell you a secret. The following secret is quite mad because we've never done this before, right? Our exam season package is normally £149, okay? But today we've got the following code, Livestream30 for £30 off. Abdurrahim, is the link in the chat, yeah? Because I can't see it. Because I don't have the chat up. Right, he's got his headphones in first. Is it in, yeah? Okay, cool. So guys, let me show you what this does, okay? Let me show you what this does. Let me put my screen up now for you guys. If you go to the following page, okay, if you go to the following page, then you do the following. So I'm on this page now, examqa.card.co, not examqa.com. Go to this page if you want to get the live stream code, right? You're going to go to this page. You're going to scroll down to the package, okay? Get full access. It's normally, look at that, it's actually normally £149 for the annual package. But for exam season, we brought it down to £59. But we know that you guys have exams so soon. So is it, you know, we want to do something special for you guys. If you put this code in, okay, so I'm going to click, you know, check out. If you want to buy a webinar, you can get that on top because, of course, you've got so many exams coming up. But I'm going to click no thank you for now. I'm going to go to the checkout page here. So it's £59. Add promotion code. And I'm going to write live stream 30. Apply. And bang. That's £29 there. Okay. When you check out, the following is going to happen. Okay. You're going to get access to Examqa. What is Examqa? All the slides that Mohammed is using right now, okay, you can use for yourself, okay? So if you feel like he's going too fast and whatever, right? It doesn't matter. It's an afternoon exam, so you can take what topics you want to go through at your pace. And we've sectioned it so nice. Look, paper 1 content, you can go through these slides. Scroll down. Paper 1 content, you can go through the slides. These are slides that we've put years of teaching and expertise and, you know, really hours and hours of behind the scenes work into these slides. And if you check the live streams from the previous years or you ask the people from the previous years, they will tell you how much of an amazing resource this is. People went from grade 2s to grade 5s. People went from grade 3s to grade 8s and 9s. And it's from these slides.
What, is he giving them £30 off?
Yeah, £30 off. Normally it's £10 off, but this year we thought, you know, we're going to do something special.
So might have to close the centre after that.
So guys, this is it. Look, you click the slides and exactly how Mohammed was going through it today. Yeah.
Why have you been so generous?
You can go like that. Click, 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 click. It's the same stuff.
Well, I'm about to teach that to him.
Oh, okay. Let me get this
fair
rid of this,
enough,
'cause
okay.
they need to buy that. So guys, I hope that makes sense. Click the website in the chat that our moderator put on and you can get this discount. The code Livestream30. After this live, Mohammed is back now, he's going to teach. But after this live, we might discontinue it. Woo. So only tonight, basically, you can use this. So, I mean, it's up to you guys. You know, you can learn the entire spec in just one night. This is proud, this is proven. It's up to you guys. I mean, fails me, it's a no-brainer because my science is not that good. But Mohammed, off to you.
Okay, that's...
There you go.
Okay, Zishan, thank you very much for that. Guys, listen to me. I just remembered something, but I actually forgot it. I was gonna say, maybe it'll come to me. How did I forget it that quickly? Okay. Anyway, it'll probably come back to me. One sec, everyone. 1 sec. So we've done that. Let's keep going, guys. You guys are doing very well so far. Oh yeah, what I was gonna say. Basically, after you watch this live stream, yeah, I want you to go to sleep. And then wake up early tomorrow, maybe an hour earlier than you normally wake up, and watch it again. And then go to your exam. That's what a lot of students did last year, and the information stayed in their brain so well. One second. Okay, ready, next one. Pretty well so far. Oops. Here we go. Okay, so guys, we're going to move on to the next topic, and that topic is to do with drug development. Yeah, here we go. All right, so first of all, what is a drug? A drug is a chemical that changes how the body works. Yeah, so a drug is basically something that changes. Now, a drug could be caffeine is a drug. For example, Red Bull makes you more alert. Alcohol is a drug, or you can have other drugs such as painkillers. Okay. Antibiotics, etc. Now, one thing with antibiotics, remember that they only kill bacteria, yeah? They don't kill viruses. So to kill viruses, we use antiviral drugs, yeah? Okay, so antibiotics are for bacteria, antivirals are for viruses. Now, very quickly, we're going to look at something called antibiotic resistance, which is basically how bacteria become stronger over time. So let's look at this example. Number one, let's say a person becomes infected with bacteria, okay? So you have a bunch of bacteria inside their body, and you can see all these bacteria are making this person ill. So the person takes some antibiotics. Now, what the antibiotic will do is it will kill some of the bacteria. Now, it's not going to kill all of them straight away because he has a couple of days of antibiotics to take, but maybe after a couple of days, maybe after 1 or 2 days, the person feels better because the majority of the bacteria have been killed, okay? Now, at this point, he starts to feel better and says, oh, you know what? I know the doctor told me to take it for 5 days, but I feel better on day 3. So I'm not going to take the remaining antibiotics. This is a big mistake because the remaining bacteria, although they're few in number, they're strong. So what's going to happen is they're going to multiply and they're going to get larger in number. So now the person starts to feel ill again. So he goes back to the same antibiotics, but guess what? The antibiotic that he used is not going to work anymore, okay? What is this called? This is called antibiotic resistance. Now, how do we prevent this from happening? 2 ways. Number one, first of all, how do you treat this person? You can either give him a stronger dose, okay? Or you give him a different antibiotic, but you can't use the same one. So how do you stop antibiotic resistance from happening? 2 ways. Number one, you should always finish the course of antibiotics. So if you don't finish the course, it means the strong bacteria that remain in the body are going to come back, and they're going to come back stronger. So number one, always finish the course. And number two, doctors should not prescribe antibiotics for things that are very simple. Okay, so for a very mild illness, don't prescribe antibiotics. Your own immune system can fight it off. Okay, let's keep going. Now, we're going to quickly talk about where drugs come from, okay? So drugs come from plants. So for example, aspirin is a very important drug. It's a painkiller. It comes from the willow tree, okay? Digitalis is a drug that helps with heart conditions, and that comes from the foxglove plant, okay? We also have drugs that come from microorganisms. For example, penicillin is an antibiotic, and it comes from fungus, okay? Etc. Now, the last place that drugs come from are from the pharmaceutical industry. So these are medicines that are made by scientists to help cure all sorts of illnesses. Okay, so let's talk about that very quickly. So here we go. Ready? Now, there are 3 stages in developing a new drug that we have to be aware of. The first stage is called, basically, all you're going to do is very simple. We're just testing the drug on cells and organs. Okay, so stage 1, very easy. Test the drug on cells and organs. Yeah? If it passes stage 1, then we move on to stage 2. Okay, stage 2 is where we start to test the drug on small animals. And what we're looking for here is we're looking for efficacy, which means does the drug work, and toxicity, is it dangerous? And if the drug passes this stage, then we go to stage number 3, where, oh, by the way, so one thing I forgot to mention, in stage 2, sometimes you might have ethical issues. So for example, they might say, oh, you shouldn't use little animals to test out drugs because you hurt them. It's true. So what we do is we usually test it out on animals that are, for example, let's say you have 2 options. Test it on a tiger or test it on a rat. And the question is, why would you pick a rat instead of a tiger? Well, number one, rats are much larger in quantity. Okay. Number two, rats are much more easier to handle. And number three, rats are much more easy to obtain. Okay. So those are some of the reasons, like, you might pick one animal over another. Regardless, there's always ethical issues. And finally, stage 3. So if it passes stage 1, which is testing on cells and organs, if the drug passes stage 2, which is testing on small animals, then we move it to stage 3, where we test it on human beings. Stage 3, by the way, has a special name. It's called clinical trials. Okay, so that's when now the drug is tested on actual human beings. Now, when we do clinical trials, we have to always use healthy volunteers. We don't use it on ill people because it might make it even worse for them. Yeah? All right. So when we do stage 3, when we do clinical trials, we test, what's the main thing that we're looking for? We're looking for side effects and optimum dose. Okay, everyone? So not efficacy, not toxicity, side effects, and what's the perfect amount of dose to give the person. Okay. Now with clinical trials, we have something called a blind trial. So in a blind trial, the patient doesn't know what drug they're getting. They could be either getting the real drug or they could be getting a placebo, which is basically a sugar pill. Okay, so in a blind trial, the patient doesn't know. They might be getting the real drug or they might be getting a placebo, but the doctor does know. And the point of a blind trial is to prevent something called psychological effects. Yeah, so in other words, the placebo effect. That's when someone takes a pill and they think they're getting better, but actually they're not. It's just all in their head. Okay, so it's to prevent that from happening. Then we have something called a double-blind trial, where the patient doesn't know what drug they're getting and the doctor doesn't know who's getting what drug. And the point of this one is to prevent bias when analyzing the data. Okay, so this prevents bias when the data is being analyzed. There you go, guys. Drug development complete. Let's go, guys. Next topic. Here we go. Anyone have a specific topic request? We'll do that after I go for my next one. Okay. Almost finished with combined science. Get ready. Triple science. We're gonna be starting very soon. Okay. Here we go. Here we go. All right. Here's a quick one. So we're gonna quickly talk about what the white blood cells in our immune system do to destroy pathogens that enter the blood. Okay. So number one, white blood cells, they do something called phagocytosis, which means that they bind to the pathogen. You have to make sure you say the word bind. Okay. They bind to the pathogen and they recognize the foreign antigen on its surface. Okay. Once they recognize the foreign antigen, they will then realize, okay, this guy's not from my body. So the white blood cell will engulf it. Okay, it will engulf and digest the pathogen. That is called phagocytosis. Number two, the white blood cell can produce a protein called antibody. So the antibody is produced by the white blood cell and it destroys the pathogen. And number three, the white blood cell also produces something called antitoxins, which removes the harmful chemicals that was left behind by the pathogen. Okay, so number one, phagocytosis. Number two, antibodies. And number three, antitoxins. Okay, good stuff. Now, very quickly, let's talk about how vaccinations work. So in the vaccination, you inject the person with a dead or weakened pathogen. Yeah? Now, even though it's dead or weakened, the antigens are still there. Okay? So the foreign antigens from that pathogen will activate the immune system of the person. Then the white blood cells will destroy that pathogen. And then they will remember that for next time. So they will turn into memory cells. So if that person becomes reinfected with the same pathogen, okay, the memory cells will rapidly destroy that pathogen before the person can even feel ill. But remember, it only works if the same pathogen enters. If the pathogen mutates, then it's not going to work. Okay, so make sure you screenshot that 5-step answer for how a vaccine works. Okay, let's move on to the next topic. One sec. Okay, guys, we're nearly finished. Okay, combined science, stay with me. We're nearly finished. I've got an amazing practical for you with a summary at the end of it, and then after that, we're gonna I'll let you guys pick the topic. Okay, here we go. So, one sec. Okay, we're going to go through a very important practical that's to do with enzymes. Okay, so here we go. Now, we know that the rate of an enzyme can be affected by temperature or by pH. Yeah, so what we're going to do is we're going to show that temperature affects the speed of an enzyme, and we're going to show that practically. Okay, so the practical that we're going to use is the reaction where starch breaks down into maltose. Yeah, now which enzyme breaks down starch into maltose? The enzyme amylase. So we're going to show that by changing the temperature of the reaction, amylase is going to break down starch into maltose much more quickly. Okay, now how do we know that the reaction is even happening? The way we know that is because there's going to be a color change. So when we have starch in our test tube, when you mix iodine with starch, okay, the test tube is going to be blue-black. The solution will be blue-black. But as the starch breaks down into maltose, the color is going to turn browny orange. Okay, so all we're going to do is we're going to time how quickly it goes from blue-black to browny orange. And remember, the quicker it changes color, the faster the reaction has happened. Okay, so here's how we do that. Number one, we're going to obviously, and at the same time, we're going to repeat this experiment at different temperatures, and we're going to record which temperature had the shortest time change. Had the shortest time for color change. Remember, the shorter the time, the quicker the reaction. Okay, so look, here's what we're going to do. So step one, we're going to, and guys, remember, you don't have to copy this out, okay? I've put a summary at the end of this. Just listen and learn, okay? Here we go. Step one, we're going to take some starch, which is the reactant, and we're going to take some amylase, and we're going to put them both into separate boiling tubes, okay? Then we're going to put them into a water bath and make sure that they're both at the same temperature, okay? So make sure that both of them are at the same temperature. Then, so for example, we'll set the temperature to 20 degrees Celsius and leave them both in for 5 minutes and make sure that they have the same temperature, okay? Then we're going to mix the amylase with the starch. We're going to mix them together. Now, as soon as we do that, what's going to happen? The reaction is going to start, right? But remember, we need to see the reaction. So what do we add? What's one more thing we have to add? Iodine. Okay, so you add some iodine solution, and because you already have starch in there, the iodine is going to go straight away blue-black. Okay, so this is the start of our experiment. We have starch, amylase, and iodine, and it's blue-black. Okay, now we have the time, how long it will take for this to go browny orange. So what we're going to do is we're going to start a timer, okay, and we're going to grab a pipette and transfer from here into the spotting tile every 10 seconds, like this. Look, 10. Then after another 10 seconds, after another 10 seconds, and you keep doing that every 10 seconds until the color is now browny orange, okay? So what does that mean? It means it took 60 seconds. It took 60 seconds for that reaction to happen at the temperature of 20 degrees Celsius. So what we're going to do now is we're going to repeat that whole experiment, but we're going to change the temperature and then see how long that will take, okay? So for example, let's say here's our data. So at the 20 degrees Celsius temperature, it took 60 seconds for it to go from blue-black to browny orange, okay? At 30 degrees, it took 40 seconds. What does that tell us? It tells us it took less time. So at 30 degrees, the reaction is happening quicker, okay? At 40 degrees, it took even less time than that, okay? Now at 50 degrees, there's no time for color change. That means the color change never happened at 50 degrees. Why? How come? It should be quicker, right? How come the color didn't change? Because most likely this temperature was so high that the enzyme denatured. And because it denatured, the reaction couldn't even happen, okay? So that's that. And so if they ask you from here, what's the optimum temperature? The optimum temperature is 40 degrees because that's the one where the reaction happened the quickest because it had the shortest time, okay? And over here, we can just say that it was very, very slow. And over here, we can just say that the enzyme got denatured. Okay, everyone? So some control variables for this experiment. Three control variables. Number one, make sure that the volume of amylase and starch is the same every time. Number two is make sure that the concentration of amylase and starch is the same. And finally, make sure that the pH, because we're changing temperature, we have to make sure that the pH is the same every time. And to make sure that's the same, we use something called a buffer solution, okay? So here's a summary, guys. Ready? Copy this out. And while you copy it out, I'm going to tell you something, okay? In this practical, remember, we were testing the effect of temperature, right, on the rate of reaction. But sometimes they could flip it around. They could say to you, oh, let's test the effect of pH on the rate of reaction. So if they ask you to investigate the effect of pH, this is what you do. Same thing, here's what you write, okay, ready? This is what you write. So I'll go back so you can screenshot it. This is for the one for temperature, okay? So if they ask you, oh, prove that temperature affects the rate of reaction. This is what you write down, okay? And if they say, prove that pH affects the rate of reaction, this is what you write down. Okay, guys? The video is recorded, so you can always go back and screenshot that. But that's your model answer that you just have to copy. Okay, I'll go back one more time in case you guys want to screenshot it right now. There you go. 5, 4, 3, 2, 1. Done. And 5, 4, 3, 2, 1. Done. Okay, banging. All right, guys, let's go for... okay, now I'm going to read some of your comments to see what topics you guys want me to go through. So if there's any topic that I haven't done yet that you want me to teach you, put it in the comments and we'll go over it. Let's go.
Before combined leave, can we just chat to them one more time for 1 minute?
Yeah, calm. Let's calm. Okay, so let me see. Guys, anyone that's saying triple science topics, we're gonna do that after combined science finish. Okay, so what's it called? Antibodies, all that. Not antibodies, monoclonal antibodies. We're gonna do that afterwards. Yeah. So don't worry about that. Meiosis. Okay. Cardiovascular system. We've done that. Okay. If some of you have joined the live stream recently, some of the topics that you mentioned, we've already done it at the start of the live stream. Okay, so when this video finishes and we upload the full live stream, you can always go back. I'll put the timestamps for you. Okay, I'm just checking if there's any topics that we haven't. Okay. All right, fine. Someone said lungs. Okay, let's quickly do lungs. Yeah. For lungs, guys. Okay, lungs will be a quick one. Don't worry. Here we go. 2 minutes on the lungs. Let's go. So we know that the lungs move oxygen into the body, right? They move oxygen into your body, and they also move out carbon dioxide out of the body. Yeah, that's what they do. But why are they so good at doing this? Here are 3 reasons why. Number 1. So, very quick reminder of the structure of the lungs. So you have the trachea, which is the windpipe, okay? You have the 2 main branches, which is called one is called a bronchus. 2 of them are called bronchi, okay? So you got trachea, you got the bronchi. Then you have the smaller branches called the bronchioles, and at the end of those bronchioles, we have the tiny air sacs called the alveoli, okay? So that's the structure of your lungs. Now, around the lungs, you have the rib cage, which is there to protect the lungs. And between the ribs, you have a muscle called intercostal muscles. And those muscles help to squeeze the lungs, to help squeeze air out of your body, and also to bring air into your body. Okay? Now, by the way, the intercostal muscles and the diaphragm work together to squeeze your lungs, so that way you can breathe. Okay? So those are the 2 main muscles. Now, let's quickly go to... the alveoli, yeah? Now, the alveoli are the tiny air sacs at the end of your lungs, and this is the site of gas exchange, okay? That's where gas leaves the gas exchanges between your lungs and your blood vessels. But why are they so good at doing gas exchange? Here are 4 reasons why they're so good. Number 1, okay? They have thin walls. The alveoli are 1 cell thick, okay? So that means it reduces the diffusion distance. So for each adaptation, you have to also write the explanation. So alveoli have thin walls, which reduces diffusion distance. Okay. Number 2, they have a good blood supply. What does that mean? It means you have capillaries surrounding the alveoli. Okay. There's capillaries surrounding it, so there's blood always circulating around the alveoli. So how does that help us with gas exchange? Well, it means that there's always a concentration gradient. That means that the moment oxygen comes in from the air, it goes into your lungs, it moves into the blood, and it's taken away so that more oxygen can come in. Okay, so the explanation is maintains a concentration gradient. Number 3, they have moist lining, which means that it helps to dissolve gases, okay? And number 4, you have millions of alveoli. So that way, it produces a large surface area. In fact, I'm not sure if this is the correct statistic, but if you take every single alveoli from your lungs and you cut them open and you lay them out flat, it's the size of half a tennis court, okay? So we have so many alveoli, it creates a massive surface area for gas exchange. So these 4 points that you guys see on the screen, make sure that you remember them. That's why, and basically that's for gas exchange, okay? And the lungs, done, good. Give me another topic, guys. Give me another topic. Give me another topic. We've done osmosis. Okay, let's have a look. Okay, someone said cardiovascular. All right, fine. We'll do cardiovascular very quickly. Cardiovascular disease. Well, it's going to be a very quick one, okay? Don't worry. It's going to be very, very quick. One sec. We'll quickly do cardiovascular, and then we'll do your next part. Potometer, we've done that one. Okay, fine. You know what? Let me quickly do. One sec. We'll do potometer, okay? That's the important one. Alright guys, we're going to quickly do this one. This one's actually more important. Ready? Factors affecting transpiration. Here we go. Lock in, guys. Let's go. Let me drink my Red Bull. Let me sniff my ashwagandha. Alright, here we go. Let's lock in. So, we're going to look at the factors affecting transpiration. Here we go. First of all, what is transpiration? Transpiration is the movement of water through a plant, okay? Now remember, water evaporates from the leaf, okay? Evaporates from the leaf and it diffuses out of the leaf. Because of that, it creates tension inside the plant, okay? So that means more water is drawn up by the xylem to replace it. So whenever water evaporates and diffuses out of the leaf, more water goes up the plant through the xylem, okay? And then finally, this whole thing is called a transpiration stream. And at the bottom, water enters the root by osmosis, okay? So here are the 4 parts of transpiration that we have to know. So in summary, water leaves at the top, it diffuses and evaporates, okay? That makes more water go up the plant through a transpiration stream. And then at the bottom, at the roots, water enters by osmosis. But the rate of transpiration, in other words, how quickly water goes through the plant, can be affected by several factors, okay? So to investigate that, we're going to do an experiment called the potometer. The potometer is a practical to investigate the rate of transpiration, to see how quickly water is passing through the plant, okay? So what we have here is we have a capillary tube, we have an air bubble over here, okay? And what's going to happen is, as water leaves the plant by transpiration, this air bubble is also going to move along the capillary tube. And the further it moves along, okay, the faster the rate of transpiration. Now remember, when you do this experiment, it's very important that it is airtight, which means no other bubbles can enter, and it's watertight. There are no leaks. Very, very important part, okay? So, when you do the experiment, you have to also make sure that the tap is closed, yeah? So that way no water goes down the reservoir. And I'll tell you about the reservoir in a second, what that's there for, okay? So, we're going to, basically, what we're going to do is we're going to measure the distance moved by the capillary tube. But at the same time, we're also going to change the factors around the plant. So, for example, let's talk about some factors, yeah? Here we go, ready? One sec. Here we go. So 4 variables that affect the rate of transpiration. Number 1, temperature, okay? So as temperature increases, particles have more kinetic energy. So instead of particles, you can say water particles, yeah? Have more kinetic energy. So there's going to be a faster rate of transpiration. So the bubble will move faster as the temperature increases. Okay, next factor, sunlight. As it gets brighter, more stomata open. Okay, and again, if there's more stomata opening, it means that the air bubble can travel further along because there's going to be more transpiration. So that's the second factor. What was the first factor? Temperature. What's the second factor? Light intensity. The third factor is humid. Oh, by the way, one more thing. When we're doing the light intensity practical, always remember we use an LED bulb. Don't use a filament lamp. Why? Because LED gives light without heat. Whereas if you use a filament lamp or a standard bulb, it will also produce heat, and then your experiment will be flawed. Okay. Third factor, humidity. What does humidity mean? It means how much air is already, how much water vapor is already in the surroundings. Okay. So if there's already a lot of water vapor in the surroundings, will water leave the plant? If there's already a lot of, if it's very humid, will water leave the plant? No, exactly. So the rate of photosynthesis is going to be high or low? Low, exactly. Okay, so when humidity is high, when it's very humid, then there's too much water vapor already in the air. So water from the plant will not leave and evaporate. So the rate of transpiration will drop. Okay, so that's what you say. You say that the amount of water inside and outside is similar. Because it's similar, there is no concentration gradient for diffusion to occur. Okay, and that's why the rate of transpiration is low when it's very humid. Okay, last one. Windy. So when it's very windy, what happens? Water vapor that's already around the plant is going to be pushed away. Okay, as it gets pushed away, that means more water will be forced to leave the plant by transpiration. So the bubble is going to move along faster. Yeah. So that's so basically when it's windy, there's going to be more transpiration and more water inside the plant will be pushed out. Okay, so the rate of transpiration increases. There we go. So that's the 4 factors that affect the rate of transpiration, and we've done that using a potometer experiment. Good stuff. Okay, so before we start triple science, which we're going to start very shortly, Zishan just wanted to say one more thing, and then after that we're going to start triple science. So lock in, guys. Get ready.
Guys, just final message, especially the combined science guys. Of course, during the live, you need to lock in. But like we mentioned, one-time offer only. Code Livestream 30 for £30 off. You get all of the slides that Mohammed has been using. One sec. Abdurrahim. Send it now, yeah. I disabled the chat. Send it. So, guys, if you check the comments right now, this page has got the exam season package. On this page only, you can use the code. Okay, so I'm going to go to that page. This examqa.card.co. Okay, so this page only, which you're seeing right now on the YouTube live, you can have access to all of the slides Mohammed is using. Again, you might ask yourself, what's the point? Your exam is tomorrow, right? You still have the rest of all your other science exams. You've got chemistry paper 1 and 2. You've got physics paper 1 and 2. What do you think, our platform has? It's got the exact same slides that Mohammed has been using, but we've got it for chem and physics as well. And arguably, in my opinion, I think chem and physics is a bit harder than bio. Okay, so if you found this live stream useful, think about if you had all these slides to yourself. Yeah, you don't need to revise the day before. You can revise them now. Physics is a bit later, chemistry is a bit later. And if you buy the package, I'm just going to show you this now. You get the platform which has the resources that Mohammed has been using. Okay, so I hope you guys have been using this live productively and usefully. Just one more sneak peek of what it's going to look like. Bang, you get access to all of this. GCSE Chem, Bio and Physics, okay? That's why it's worth it. Don't think, oh, my exam is tomorrow, what's the point? You got more than just Bio, mate. You got Bio Paper 2, you got all these exams, okay? If you want to use the slides that Mohammed's been using for yourself, not in this fast-paced method, which unfortunately we have to do because it's the night before, then use that discount code that our admin has put in the chat. £30 off, it becomes only £29 when it's usually £149. Thanks so much, guys. I'll leave it back to Mohammed.
Let's do it.
Buy the slides. Get the slides.
Okay, nice. All right, triple science. Here we go, guys. Time for triple science. Combined science. We'll put the we'll put the timestamps after the live stream, okay? So you can watch it tomorrow morning one more time before you go to your exam. But now we're gonna quickly go through the triple science topics. So let's do it, guys. Here we go.
Combined Science can watch the live after it's been posted as well.
Yeah, Combined Science, the live is gonna be posted straight once as soon as this finishes, okay? What's this thing on the screen? Can I delete that?
Don't delete it, but one second.
All right, nice. Here we go. Okay, triple. Okay, first one, binary fission. Okay, so we know that prokaryotic cells are a type of cell. Okay, they don't have a nucleus, but they do have genetic material, and that comes in two forms. It has chromosomal DNA. Okay, bacteria, for example, have chromosomal DNA, which controls the activities of the cell, but they also have circular rings or circular DNA called plasmid. Okay, now what do plasmids do? They contain genes for antibiotic resistance. That's what plasmids do. Now bacteria do not divide by mitosis. Instead, they divide by a different process called binary fission. Here's how it works. Number one, the plasmid and the chromosomal DNA replicate. That's step one. Okay, but step two is while the plasmid doubles, so as you can see, while the chromosomal DNA doubles, the plasmid multiplies many times. So it could be 4, 5, 6, or 7. Yeah. Now, after that happens, the bacteria divides. Now, in binary fission, the two daughter cells that are produced are not identical. Even though they both each have one copy of the chromosomal DNA, they have different number of plasmids. And that's why, unlike mitosis, which produces identical cells, binary fission does not produce identical cells. Okay, now let's do a quick experiment. We're going to investigate how well an antibiotic can kill bacterial cells. And to do that, we're going to do the following. Step one is so we have a bacterial culture over here, and we're going to use these we're going to grow them, and then we're going to kill them. And then we're going to see which ones survive. And those guys will be the champions, okay? So step one, you're going to sterilize an inoculating loop. So you're going to get a loop of platinum wire. You're going to pass it over a Bunsen burner, yeah? Because you want to kill any existing bacteria on there. Then, once you remove it, you let it cool down. Right now it's going to be very hot. You let it cool down, and then you grab the bacteria from the culture. Okay, so you get a bunch of bacteria, and then you spread that bacteria on a petri dish, which has some nutrients for the bacteria to grow on. Okay, so you spread all that bacteria onto the petri dish, and yeah. Now, while you're doing that, you also get some paper discs, and you put them into different types of antibiotics. Antibiotic A, B, C, and also in water. Yeah? So you put them in here. Now, why water? Why do we even use water? Water is the control, okay? We're just doing that to prove that our experiment works. Then you remove them, okay? And then you put them into a petri dish. So those paper discs which had antibiotics, you put them onto the petri dish at different positions, and then you're going to place a lid on the dish and cross tape it. Now, why do we cross tape? Why don't we just do all the way around? Why do we cross tape it? It's because we want to allow oxygen to still be able to go through and allow the bacteria to try to grow from that. Okay. Now, at the same time, once you cross tape it, you flip the petri dish upside down. This stops condensation. Okay. You leave it at room temperature so the bacteria can grow, and you leave it for a short interval of, let's say, 48 hours. Now, when you come back the next day, this is what you're going to see. When you open the petri dish, you're going to see the following. So you have these paper discs, and around them, we have these clay zones. Can you see? Look, there's a clay zone here, there's a clay zone here, and this one's got a tiny one. Those clay zones are called inhibition zones. It's the areas where the antibiotic did not allow bacteria to grow. In other words, these are areas where bacteria were not able to grow. And the bigger the inhibition zone, okay, the bigger, the stronger the antibiotic. So in this case, which one is the strongest antibiotic? This one over here. That's the strongest because it had the largest inhibition zone. Okay, everyone? So, one second. So, yeah. Yeah, this is triple only. Okay, so the bigger the antibiotic, the bigger the inhibition zone, the stronger the antibiotic. All right, moving on. How do we make sure? And of course, remember with the water one, the control experiment, there should be no inhibition zone, right? Because water is not an antibiotic. It does not have any effect on bacteria. So look, there's no inhibition zone here, okay? And if there was, that would be a mistake. But the fact that there isn't, that's good. That proves that your practical is valid, all right? So here's a summary of how to do this practical, how to investigate the effect of antibiotics on bacteria. One more thing, to make sure our results are reliable, here are 4 things that we have to keep constant. First one is, all the antibiotics should have the same concentration, okay? Easy one. Second one, the paper discs should have the same size, thickness, and be made of the same material. Number 3, you want to do this experiment in sterile conditions, so you want to wash your hands and clean the surfaces. And finally, you want to make sure that the paper discs are placed evenly apart and there is no overlapping. Okay, so those are 4 things you want to do. So 1, 2, 3, 4. 4 things that you want to do to make sure that your results are reliable. Okay, everyone? Smashed it. That's the first topic. Done. Let's move on to the next triple science topic. All right, here we go. Next topic. Guys, remember, if I went through this very quickly, don't worry, you can revisit it again when you watch the recording. All right, let's go to triple science. So we've got all right, let's go through monoclonal antibodies. Ready? Here we go. So first of all, there's a special cell in there's a special type of white blood cell called a B lymphocyte, and this white blood cell is responsible for making antibodies. Okay, that's the only one that makes antibodies. Now, what are antibodies? Antibodies are proteins that have a specific shape, and they bind to antigens on the surface of pathogens. Now, imagine this. Here I have a bunch of antibodies, okay, but they all have different shapes. So this group of antibodies is called polyclonal, yeah? They have different shapes. What I want is not this. I want a group of antibodies that are all the same shape. That's what we refer to when we say monoclonal antibodies, okay? So you want all of these antibodies and we want them all to have the exact same shape as each other. So how do we get this? So step 1 is the following. Step 1, we're going to inject an animal like a mouse with a specific antigen that we want. Okay, so step 1, inject an animal with an antigen. Why do we inject it with an antigen? Because that will activate the B lymphocytes in the immune system of the animal. So now the B lymphocytes in the mouse's immune system will start to produce antibodies against the antigen that you just injected it with. Okay. Then you remove the B lymphocyte from the mouse. Okay, you remove the B lymphocyte from the mouse, and then what you're going to do is you're going to take a tumor cell. What's a tumor cell? It's a special cell that divides rapidly. Okay, it doesn't make antibodies by itself. It's just very good at dividing rapidly. And then you're going to fuse them together. So the tumor cell that divides rapidly and the B lymphocyte that makes antibodies, you're going to fuse them together, and that's going to produce something called a hybridoma. So what is a hybridoma? It's a cell that divides rapidly, plus it can make antibodies. Okay, so now what you're going to do is you're going to allow the hybridoma to produce a bunch of antibodies for you very, very rapidly. And then there you go. So you have lots of hybridomas. They're going to make the antibodies for you, and then all you have to do is extract the antibodies, and there you go. You have your monoclonal antibodies all ready and collected. Okay? Now, what do we use monoclonal antibodies for? We use them for a bunch of stuff. The first example is we use them for cancer treatment. Why? Because cancer cells, they have different antigens to healthy cells. Okay, so what that means is if I make a monoclonal antibody that only binds to the antigen of a cancer cell, then I can give that antibody an anti-cancer drug. So it will only bind to the cancer cells and not to your healthy cells. So that way you're not going to have any nasty side effects, because the antibody is only targeting the antigens on the cancer cells and not the healthy cells. Okay, The second use of antibodies is in pregnancy tests. So for example, let me show you this example, okay? Here we have a pregnancy test strip. On one side, we have monoclonal antibodies, okay? Again, they all have the same shape, and they are not stuck to the strip, okay? They're there. What they do have is they have blue beads attached to them. So you have antibodies and blue beads on one side of the strip, okay? On the other side, you have the same antibodies that are stuck down physically. And these ones, they don't have the blue beads, okay? Now, let's say that the lady wants to test if she's pregnant. So what she's going to do is she's going to, if she is pregnant, in her urine, there'll be a hormone called Hcg, okay? So the pregnant lady is going to urinate on this side, okay? And what will happen is, that hormone, Hcg, will bind to the antibody that's on the strip. Okay, so you're going to have hormone, antibody, and then you're going to have the blue bead. Okay, so you can urinate there. It's going to look like this. Now, because this area is wet, the whole antibody, blue bead, and hormone combination is going to travel along the strip. Okay, it's going to travel along the strip. Once it gets to the other side, these hormones will bind to those antibodies, okay? So the antibodies that are stuck will bind to the hormone and the blue bead. So this whole area will go blue, okay? And that means, yes, you're pregnant. Now, if the lady is not pregnant, when she urinates on this side, is there going to be any hormone released? No. So when the antibodies move along, when they travel along the strip, what's going to happen? Do you think that these antibodies and blue beads will be able to attach to these antibodies that are stuck down or not? No, they're not, right? Because there's no hormone to bridge the gap. So they're just going to fall across, they're going to fall off the strip, and it's not going to go blue. And that means that the lady was not pregnant. Okay, guys? That's how the pregnancy test strip works. Good stuff. All right, nice, nice, nice. What is next topic? Okay, let's quickly go through. Okay, guys, we're going to very, very quickly go through plant diseases, okay? Here we go. Plant disease and defense. So, first of all, let's quickly go through plant disease, okay? So, plants need minerals from the soil in order to grow. What are the minerals? Nitrates. Nitrates are there for proteins, for growth, okay? If they don't have nitrates, they're going to be small. They're going to have stunted growth. Okay, next one. Magnesium is to make chlorophyll, which gives it that green color. So if there's no magnesium, no chlorophyll, no green. Instead, the plant will go yellow, yeah? So lack of nitrates, no protein, no growth. Lack of magnesium, no chlorophyll, yellow leaves, yeah? Okay, next one. If a plant becomes infected by pathogens, just like us, we get infected by bacteria, plants can also become infected. Okay, so how do you know if a plant's infected? Here we go. So, number one, it will have stunted growth. It will be smaller than usual. Number two, it will have spots on the leaves. Number three, it might have patches of decay, areas of it that look like it's been decayed, okay? And finally, it will have discoloration on the leaves. These are 4 signs that the plant has, is undergoing sort of disease, okay? So, sorry, the last one I forgot to say, malformed stems. So just make sure you remember all 5 of these. Next part. How do plants defend themselves? So just like we have an immune system, plants also have a defense mechanism. So let me show you. The first one. The first one is the waxy cuticle, which acts like a barrier, okay? So that's just the very basic. Just like we have skin, plants have a waxy cuticle that acts like a barrier. Number 2, plants have cell walls made of cellulose. Again, thick, makes it difficult for bacteria to get, or sorry, not bacteria, makes it difficult for pathogens to get inside the plant, okay? And finally, they have bark around the stem. Bark is made of dead cells, but it's a thick layer. Just like the waxy cuticle, it protects the plant. Okay, so those are the physical defenses. But plants have other special defenses as well. Not just physical, they have a couple of other ones. Let me show you. Here we go. Ready? Oops, one sec. They also have chemical defenses. So, what's an example? Plants can make chemicals that kill bacteria, okay? And also, plants have poisons which taste bitter, so an animal won't try to eat it again. And the last one, so we spoke about physical, we spoke about chemical. The last type of defense that plants have is called mechanical. Now, let me give an example, okay? One example of mechanical defense is thorns and hairs, which stops the animals from touching them. Sometimes you might see a very nice rose, but it has thorns on it, right? That's because the rose doesn't want you to touch it. It's like, go away, don't touch me, yeah? I've got thorns on myself. The second one is that sometimes plants, when you touch them, they suddenly curl up and they hide like a tortoise, okay? That's, again, the reason it's doing that is to knock off insects. And the last one is they do something called mimicry, where they look like an animal. So I don't know if you've seen those pictures of plants that look like animals. Google it, monkey plant. You see that if you look at this, you might think, oh, this is a monkey. It's actually a plant, okay? It just looks like an animal to scare animals from trying to eat it, okay? And that's called mimicry. So these are 3 ways that plants defend themselves. Okay, nice. I believe that we are at the end of the live stream for Triple Science. Guys, that was the extra bit for Triple Science. Apart from that, does anyone have any specific topics? Okay. Ooh. Guys, a lot of the stuff that you're saying is coming up in paper 2, okay? So we're going to do that. Kidneys, all that's paper 2. That's all coming up in paper 2. Protein synthesis, that's coming up in paper 2 as well. Alright, what I will say, aseptic techniques, we've done that already. We just did that. Okay, fine. I'm going to... Alright, guys. Final advice from me is... Tomorrow, paper 1 is going to be the first of all your science papers. If this goes well... and you can make it go well, just make sure you wake up early and watch the live stream again. It's going to set a very good momentum for the rest of your papers. The only thing that I don't want you to do is come out the exam and feel demotivated because someone had a different answer to yourself, okay? Maybe your answer was right, maybe theirs was wrong. So just make sure that after the exam, you go straight home, start revising, and join us for paper 1 math live stream. When is that going to be, Zishan? Wednesday night, okay? So Wednesday night, 8:30 p.m. I'll see you guys. Hopefully paper 1 goes well tomorrow. Let me know after you've done your paper in the comments, and then hopefully I'll see you guys for maths paper 1. And that's good night from the team at Examqa. Take care.
Boys and girls.
Good night. Zishan.
Yeah, let me plug them hard.
Oh, okay. Zishan's gonna sing you guys lullaby. Yeah.
Unskippable mod, unskippable ad.
Lullaby.
Boys and girls, we're going to leave the chat open for a bit more because I know there's some unserious people that want to have a bit of baking off before the night before they go to sleep. Boys and girls, let me mention again something very important. I mean, again, exams, year 11 exams, they're big, they're very tough, they're very scary. But trust me, if you really digest the information that you learned tonight, you apply that tomorrow, you'll be shocked what grades you get. If I could show you, because it's a bit long to show you right now, I'll prepare it for the next live stream. But if I show you the comments people were saying last year and the years before, Mohammed, I need to buy you, when I get my first wage, I'm going to buy you Ferrari. Mohammed, when I, grow up and become a doctor, I'm going to give you a free surgery. These are comments not because they love Mohammed and, he's just so good looking. It's because he saved their bio exam, chem exam, physics exam. How did he save them? I'll show you right now. Oops, that's top secret. There, can't show you that. How did he save them? It's because of the slides. The slides are our special weapon. We've created this weapon that no other platform has created. These slides let you go from a grade 1 to a grade 7, 8, or 9. These slides let you learn the entire specification in one night. You can literally learn 30 or 40 topics in one night. Yeah, it's ridiculous. It's unfathomable. It's unspeakable. But it is how it is. Yeah. Here you go. You click the examqa.card.co, which our admins have put in the chat. Yeah. You click, it's in the description as well. If the chat is being proper bakey right now, just go to the description. It's at the top of the description. You click get full access. If you want to buy the webinar, it's basically a bunch of, it's basically a live stream, but it's a longer version of it. You can add that to your order if you want to. Yeah. You click no thanks, I'll continue to checkout, and bang. Promotion code only for today. This promotion code only for today is probably going to get deactivated tonight. You click live stream 30. You click apply. Okay, you ask your guardian or carer if they have your credit card details. If you have your own debit card, use your own debit card, no problem. And bang, £29 for a disgusting amount of resources. What do I mean by that? Bang. Look at this. It's not just slides you're getting, by the way. Let's, for example, click AQA Bio, right? Oops, need to log in. Not going to show you my login details. That's a free account that you don't deserve. What are they saying, Arba? They're saying good things.
Some guy said, I love brain and tongue.
Brain and tongue. No LARPing from that guy, please. Okay, look at that. Bang. I'm going to click. Okay, you got, what's the hardest exam? Chem or physics? Chem or physics? Write in the chat right now, biochem or physics, which one's the hardest exam? Someone write in the chat, tell me what they think the hardest exam is. I'm going to go to that platform. Has anyone said anything? Chem, okay, cool. Look at this. If you do Edexcel chemistry, I'm going to click Edexcel chemistry, yeah? I'm going to let you in another secret, by the way. This is a fake microphone. Look at that, bang. There's nothing in there. Sorry for exposing. Look at that. Paper 1 content for Edexcel chemistry. Look at this. Videos, slides, notes, worksheets, tests as well. Oh my God, what more do you want? So not only do you get slides, I'm going to click the Edexcel Chem topic 2 combine notes. Bang, summarized notes there. Okay, my iPad is a bit stretched, but on your laptop you'll be fine. Okay. Triple notes as well. Combined. Do you see how we've segmented it? It's combined and triple. There we go. That's a bit better now. It's combined and triple separated. So we've got these summarized notes. We've got these slides that you can use at your own pace. You don't have to rush it like we've been doing tonight. You can take it at your own pace. Wow, look at that. That's a disgusting slide right there. Disgusting in the good way, I mean. Both physically as well, because look, it's about poo and stuff. What is this about? Wastewater. Look, I'm not even a chemistry teacher, right? But I could teach you this right now. That's how, what is this? It's poo being built up. Wow, look at that. And then they get the actual water, and then you drink that, the sludge. Wow, look at that. I did not know that, in fact. So do you see, these slides, we've put our heart, body, and soul into them. And you can get them for only £29. I mean, I don't know what other offer you need at this moment in time for your exams, but that's the offer that, you know, we're giving. And it's only a one-time thing. Boys and girls, I don't want to keep it too long. It's already 10:20. You've got, is it a morning or afternoon exam? I'm not actually sure. I think it's I don't know what it is actually. I think it's an afternoon exam. So you can have a bit of a lie-in, but what is our suggested method? Have a good night's sleep tonight. Wake up early, yeah? When you wake up early, you can digest that information in a much more better manner. You can digest the information in a more memorable way. That long-term memory you're going to have in the exam is better than if you stay up tonight and revise. Because when you wake up, you're probably going to forget all that information. However, if you go to sleep now and wake up early in the morning, wow, that's a fresh brain you have there. Because tonight, think about it, you've been up for already about 15, 16 hours, right? If you're trying to revise now, after this live stream, all that information, it's like your brain is like a computer. You've already got so much RAM going on, random access memory. You're not going to remember all the information you're studying right now. So if you take a sleep, the RAM, the random access memory in your brain, it resets. So everything that you revise in the morning sticks way more than it would now. Okay, so that's our advice. Go to sleep, wake up early, have a good morning breakfast. What's the best breakfast to have on our exam day? What do you guys recommend? Grosh. So our admin is recommending grosh. Have that. Make that. What is that? Beans, basically.
One guy said brain and tongue. Yeah.
Brain and tongue. If you can, you know, make that in the morning. Fair enough. I don't know if you guys can cook, but you know, maybe you can. Have something good, energetic, that is going to make your brain function well. Yeah. And go into that exam, and put your heart and soul into that paper. That's our advice. Put your heart and soul into that paper. Yeah? And you won't have any regrets. Is anyone still on, even on the live?
Yeah, there's like 1,100 people.
Wow. I feel like Kai Cenat right now. 1K people. Oh, didn't he stop streaming though or something?
Yeah.
Really? 4 weeks ago.
Is Kai Cenat, is he still streaming?
Nah,
Ah, he's
he's
gone.
on some. He's on some. Like,
Rediscovering himself.
rediscovered thing with
Yeah.
his journal and that.
Ah, I see. Yeah, you know, it's good to journal, you know. In life, you should journal. I mean, I remember in school, they used to try and make us put information in a homework diary and all these things. And back in the day, you know, you'd think what's the point of it? But to be honest, it's actually good to do these things. Yeah. Okay. Is there any goys left in the chat then?
There's 1,000 of them.
Wow.
1,000 goalies.
No. What's the good?
Hey Zishan, how can someone say, Zishan, don't worry mate, I'll buy a Ferrari.
Oh, lovely stuff.
Go.
So guys, subscribe. We're trying to hit the 100K plaque. Yeah, subscribe tonight. Get us to 100K on YouTube so we can get the plaque. And this is the remaining live streams we have scheduled. So if you go on Examqa and you click the live streams on the menu, bang! Look at that. Maths paper 1 on Wednesday. Chem on. Oh, you got Chem this Sunday night.
Sunday night.
Yeah, because it's Sunday. You're trying to do it earlier. You're still trying to do 8:30.
Raw. This one got berries.
The Apple one. So look, this is the schedule, guys. Yeah. Because look, if you buy the package, look, you still got so many exams. Do you see? That's why I'm plugging it right now. I'm not trying to take the mic. I'm not trying to be the unskippable ad. I'm just trying to save your exams. Do you get it? Save my exams. This is the real save my exams. Yeah? Let me show you the physics platform as well. Why not? So look, I'm going to go to GCSE Physics AQA. How many views it got?
63K. Yeah, it's not
Right. Imagine we just did an AI slop webinar, a live stream instead. Look, this is the physics slides. I'm going to let's pick the best physics slides there is. Which one is the best physics slide? Momentum. Okay, look, say you're so confused about momentum, right? I'm going to go to physics. I'm going to go to momentum. And you can get access to this now. You don't have to wait for the live. If you buy the platform, you can get access to this now. Do you see why I'm plugging it? See, I'm not trying to just make money, right? I'm not just trying to make money. I'm trying to help you guys. So I'm going to click momentum and look at this. Bang. P equals MV. Did you even know that? Stop. Look at this. Bang. Collision. So now you have to do P equals MV for both of the objects, right? You fill that in. You get the two P's. You add them together. Bang. Bob's your uncle. Do you see? This is quality right here. I mean, I don't even know if I'm allowed to be showing you this, to be honest with you. But Mohammed's going to go through it properly. But you know, what time is it? 10:23. We're going to lock off at 10:30. So you guys have your fun right now whilst you're in the chat. And, you know, just don't send any of your Snapchats to one another. You're going to get banned. But have your fun. Let me do a bit more plugging. You know, £29 only. After tonight, we're going to deactivate the code. This is why I'm really pushing it right now, because I want you guys to be able to get it. I really tried my best to let the code be on for more than tonight, but the team has said to me, no, Zishan, it's only for tonight. If we give it for more than tonight, then the hard workers that have really put their heart and soul into making these resources, it's not worth it on their behalf. We can only do it for tonight. Yeah. So this is why I'm plugging it hard. Okay, guys. The chat is quite relatively good this year, I think. There's no one that's being a bit goy.
There's no goys in the chat this year.
There's no goys in the chat this year.
I'll buy you like Baby Johnson's oil
Oh my God.
for your birthday.
No oil jokes this year. It was very good. But guys, we've got something very interesting planned. There's going to be some entertainment maybe in the next ones, possibly. Who knows? This is a serious one. We want you guys to have the best, you know, experience right now. But later on, there might be a bit of some interesting stuff going on as well. You never know. Guys, look at this. Let me show you a few more things now. I'm going to go down to another. Wow, look at that quality stuff. I'm going to click. Combined and triple separated. So do you see these slides? These are for both combined and triple. But then each topic has triple only. So you're not going to get mixed up and revise the wrong thing. We've made it very easy and nice for you guys to revise. Apologies. We've made it very easy and nice for you guys to revise. Yeah. So if you do triple, bang, click visible light and have at it. Yeah. A whole slide just on that topic and you just bang it out. It's so straightforward. I mean, if I had this when I was in GCSE, I don't even know how many packages I would buy. Probably buy 3 just for the sake of it. Do you get what I'm trying to say? Let's look at one of one. Lenses. Let's look at lenses. Bang. Look at that. Oh my God. This is quality. This is content. I mean, look how much effort has been put into this. What's going on? Why is Mod laughing? I mean, if you don't buy this tonight, I mean, I don't even know. I can't. I'm lost for words. I'm really lost for words. Yeah. You also have access to these webinars as well. If you click the webinar, the examqa.car.co, there's a webinar you can buy as well. I'll just show you quickly what that is. So we'll go to my webinars. So these are, oh, I haven't bought them. I have to buy them. How can I access them? Okay, let's just look at it anyway. So look, so you see how this live stream, for example, was only 2 hours, right? The webinar is basically an extended version of it. So if you really want to know all of the topics and all of the content, rather than just the predicted stuff, you can buy the webinar, okay, for each of them, or you can buy all 3 webinars, a bit discounted, £20 instead. And basically, it just makes you a bit more confident going into the exam, right? Rather than using a night before live stream, okay? That's what the webinars are for. If you want to buy it, you know, it's there for you. It's discounted. Okay, but use Livestream 30 while stocks still last. Use Livestream 30 while stocks still last. I mean, I can't. I don't know how we got £30 off, but we managed to do it for this year. That's why I'm recommending it. Abdurrahim, a lot of giggling. There's a lot of giggling. How come you're giggling so much?
Oh.
Huh?
I don't know, man.
What's funny? Oh dear. Okay, lads. Let's call it a day. The mods are giggling. I don't know what's going on. We're gonna have to call it a day, lads. There's 100 people still there.
Oh, 100 people here. I can't. There's almost there's 800 guys on the flipping thing. On the YouTube one.
Guys, goodbye. Okay, Ab, do you want to give any final advice?
Final advice, yeah.
Sit down, give your last. You've got 2 minutes left. Take this seat. Take this seat and give some final. What would you say to your student? What would you say to your student if if they had exam tomorrow? What advice would you give?
If I had an exam tomorrow, what would I say? I would say, number 1, wake up. Don't be lazy. Don't be a goy. Don't wake up at like 12 p.m. right before your bio exam and do niche. Okay, let me even take the microphone. It's afternoon.
Oh.
That's what I'm saying. These men have better time.
Yeah, they're gonna oversleep.
You guys are chuds. If you wake up at 12, you're a chud. You're
What does
a
that
chud.
mean? What's chud? What is that one?
Such up, I promise you. You guys are chuds if you wake up at 12 for that bio exam. You guys are chuds. If you don't,
What should they do instead?
if you wake up at like, let's say a good time, 9. If you don't have RS, apparently some guys have RS in the morning.
Oh yeah.
Fries, yeah, fries. If you got RS, you're fries. Look, you wake up at 9, okay, and what you do is you re-watch this with the timestamps. You go through everything that you need help on, and what you do is you try to regurgitate. That might be a big word for some of you, but that's regurgitate the information.
Right.
Now, once you do that, you go through your little gaps in knowledge and everything else, and what you do is you go chest up, confident that you're gonna smash that exam, that you're gonna be the next doctor, next dentist, next engineer, who knows? You need that
Let's go.
next goy. That
Top boy.
you're gonna be the top goy. You're gonna be, in G, the G in top G means top goy. That's what you guys are gonna be.
Wow, we need to.
Okay, so you're gonna walk out, chest pumping. You're gonna smash that exam, and even if you feel not that confident, just okay, it's a thing of delusion. It's a mindset. All this is a mindset, and if you have a strong mindset, you have a strong exam. Let's go. Let's smash those exams.
Wow, brilliant. Thanks so much, Mr. Ab. We really appreciate your advice. We really appreciate your advice. Brilliant advice. I'm very happy that you gave those words to our dear beloved year 11s that are taking this exam. Guys, I don't know what else to say. I think it's time to finish. The code Livestream 30 is probably going to expire at midnight tonight. Yeah, it's probably going to expire at midnight tonight. Muawiyah Musa 306, I pay for the patch, you can't access it. If you email us and you have any issues with the access, we're active tonight and we're going to fix any issues that you have, okay? So just email us and we're going to fix it tonight. Don't worry about it, okay? So apologies if there's any issues. But guys, oh my God. What a night, what a live stream. I mean, thanks so much for tuning in. We're going to leave it as that, okay? What are the best of all sayings in the end? وآخر دعوانا أن الحمد لله رب العالمين. سبحانك اللهم وبحمدك أشهد أن لا إله إلا أنت أستغفر الله وأتوب إليه. How do I end this?
Spam Rabbi Zidni Ilma and maybe.
Oh yeah, Rabbi Zidni Ilma. Remember all the du'as. There's some good ones, that you can use. Goodbye. How do I stop this? Yeah, we're gonna post a live now. Goodbye. Rewatch it.